Hello, Dr. Cisse covering anatomy and physiology. Today we'll talk about gene regulation. We'll talk about genetic in general and gene regulation. In the last video, I presented the case of your grandmother receiving the neighbor's message. Steve came to the house, knocked at the door, and told your grandmother that Ethan, your neighbor, needs cake. Then your grandmother went to the kitchen, looked for the recipe for making cake, called you, you came and opened the book, made a copy of the recipe, and called your brother Tyron to mix all the ingredients to take it to the oven to bake it and to make the cake. In this case, we said that Steve is the stimulus and the door is a receptor. So grandma is a gene activator, is a regulatory protein, right? So grandmother went to the kitchen to open the book, the DNA, to tell you to open the book. You are the polymerase. You are an enzyme whose job is open the DNA so that you can find a specific recipe, the specific gene, so that you can make a copy of that. That copy is called mRNA. So as a polymerase, you made mRNA. You took the mRNA out of the nucleus to the cytoplasm to find tRNA, Tyron, your brother, so that Tyron can find all the ingredients and put them together. And those ingredients came together to be the dough that needs to go to the oven, the ribosome, on the rough ER to be baked to make the cake. Now, let's go into the detail of the DNA itself, of the recipe book of cell. Of the 23 pairs of the recipe book, what do they have inside? How do they work? So to illustrate that, let's pretend this is a cell, so the house, and the cell will have receptors, door. So we have a stimulus coming to a receptor to excite the receptor. And of course we have a nucleus here. Then the grandmother, who is a regulatory protein, come to get the receptor's message. And the receptor said, hey, Steve, the stimulus said to make a cake. So grandma went to the nucleus through the nuclear pore. What? The nuclear pore is just a door to the nucleus. Went into the nucleus and find the DNA. In this case, the DNA are what? The books, the recipe books. Now, we have 23 pairs of chromosomes. What does that mean? Just like we said, all the books pile up together represent the DNA in disorganized manner. And when the DNA is not organized, when the cell is not dividing, all the DNA together are called chromatin. Chromatin. What is chromatin? It's just a disorganized DNA. But when the cell is dividing, the DNA organize themselves into chromosomes. And human, we have 23 pairs of chromosomes. So 23 pairs. Now, how do we translate that into our example? 23 pairs, that means that you have 46 total. It means you have 23 on one side, 23 on the other side. How do you get those 23? We get 23 chromosomes from a mother, 23 from the father. The two together become 23 pairs of chromosomes. That means there's 10 type of chromosome. Maybe they just contain different type of information. You might have blue eyes with the father and dark skin with the mother, for example. So the DNA will have chromosomes that will have specific information, but those information will have specific codes that will determine the specific character that you're gonna have. So in this case, the books will be what? The chromosomes. We have 23 pairs, total of 46. So one of the chromosomes will have the information about the cake, and that information is the gene. In our case, it will be a page of the book. So the page of the book will be a gene on the chromosome. So what grandma did is just to find the book to make sure the book can be read. When the regulatory protein found the book, found the DNA, another protein, which is an enzyme, will come. That enzyme is polymerase. Polymerase is an enzyme, whose job is to read the code on the DNA and make a copy of it. And that copy is called mRNA, messenger RNA. That mRNA, if the protein is meant to be shipped out of the cell, the mRNA will come out of the nucleus. So this mRNA will come to the nucleus, to the cytoplasm, here, to the cytoplasm. When it comes out of the nucleus to the cytoplasm, this mRNA has the message of the DNA. Now you see that the DNA cannot move out of the nucleus. It has to stay in the nucleus. Grandmother recipe cannot move out of the kitchen. But the copy of the DNA can move out thanks to the polymerase U enzyme that will take that, make a copy and take it out of the nucleus, come to the cytoplasm. Once it gets to the cytoplasm, of course it is a copy of the DNA. You have to make now the protein. Now the process of making mRNA from DNA is called transcription. What is transcription? Transcription. Transcription is the process of transcribing, making a copy of the DNA to mRNA. Transcription. Then that mRNA has the message that needs to be used to make our protein. How is it made? The ingredients, meaning the nucleotide, the pieces of the mRNA, the pieces of the messenger, the messages in the mRNA are called nucleotide. One of you asked me before, what's the difference between DNA and RNA? One of the differences is the type of nucleotide that the DNA has and the type of nucleotide that mRNA has. For example, the nucleotide in DNA will be ATCG. But the RNA will not have a T, it will have the U, AUCG. It's just a different type of nucleotide. That's one of the differences. So that means that anytime the polymerase sees a T in the DNA, it will pretend that it's a U. So whatever corresponding will come will be A, because you understand that in DNA pairing, A always go with T, C always go with G. But if we're in the RNA, because there's no T there, the A will go with the U in the RNA, okay, in the RNA. That's why in this mRNA, we'll see A, U, C, G. But in the DNA, we'll say A, T, C, G in the same where. That's why the DNA and RNA are corresponding, or it's the same message, just different type of nucleotide between the T and the U. That's one thing. Now, when the messenger RNA is made, it has to be made into the protein now. How do you put that together? The tRNA, in this case Taiwan, will have to take the pieces of the mRNA, read them, so they can find the corresponding amino acid. In our case, amino acid will be our ingredients. For example, the flour, the egg, the sugar, the salt, the milk, each one is an amino acid carried by the tRNA. So the tRNA carries the amino acid, which is just the building block of the protein. So the tRNA will take the pieces of amino acid to take it to where? To the ribosome. The ribosome, we say, that's like the oven, the ribosome. So the ribosome will take the ribosome. So the ribosome will take the pieces of amino acid and add them together to build the protein. So amino acid is what? The building block of protein. The process of the tRNA taking mRNA to make protein from amino acid is called translation. 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 It means the tRNA is translating the messages from nucleotide to protein, from nucleotide to amino acid. So now, when the mRNA is actually made, many pieces of the mRNA and RNA actually the recipe. For example, if I make a cake, making the cake, the grandma can say when you make a cake, you have to wash your hands, you have to use a spoon and fork. Those do not go to the cake, but they're part of the procedure to make the cake. Those pieces of information will be cut out from the mRNA. That mRNA is called pre-mRNA. What does the pre mean? Before it becomes the complete mRNA. So the pre-mRNA will be just like the non-clean mRNA. So pieces are removed to clean it so that it can be complete recipe to make the cake. The pieces that are removed are called the introns. Introns. What is an intron? It's the piece of the mRNA that is removed before it can go to the ribosome to make the cake. What about the exons? The exons are the pieces that are kept. So they're expressed. That's what they call exon, right? Expressed. So in this case, the mRNA comes from the pre-mRNA because it's the first one that is made. Why? Because it has not only information to make the cake, it has also extra information to make the cake making even better. But those information do not go into the cake itself. It's not flour, it's not egg milk, it's probably the spoon you use, the fork you use, all the stuff. So those pre-mRNA become mRNA, and that mRNA is transformed into the protein. So making protein from mRNA is called what? Translation. So making protein comes from the DNA, DNA to mRNA transcription, mRNA to protein translation. All these are done because a regulatory protein, your grandmother, understood the stimulus coming from a receptor, came to the DNA to find the book, to find a specific page, and that page is called what? The gene. So the gene is the specific page of the DNA, the specific page of the book, a specific message in the DNA that will be used to make the cake, right? So at this point, we understand all the points, all the elements here, right? We know what is the stimulus, the receptor, the regulatory protein, the nuclear pore, the nucleus, the DNA, the chromosome, the gene, the polymerase, which is an enzyme, to make the mRNA that comes from the pre-mRNA, then the mRNA. And the tRNA will take the pieces, nucleotide, and call in the amino acid, which are the ingredient, take them into the ribosome to put them together to make the protein, and that protein can be secreted out of the cell to go to our neighbor Ethan, who needs to eat the cake, right? So I think, and we understand the difference between transcription and translation, right? From DNA to mRNA is transcription, from mRNA to protein is called translation. We know the difference between exon and intron. Exon will be the part of the mRNA that will actually be the cake, the protein, and the intron, your intron will be just something between them, like international, between. There's be cut out to clean the mRNA so you can talk about, so you can make specifically the cake, right? So I think everything is clear here, and I hope to see you in class. Thank you very much.